All right, hi everybody. I am back for part 2 of excitable membranes. And this side or step is depolarization. So what happens when you take a membrane that has been polarized, which means there's charge separation across the membrane, and then you undo it. So I'm going to start by drawing my membrane, like always. And then this time I'm going to draw some different types of channels. And let's draw one of these. Okay. And I'm also going to put a sodium-potassium ATPase in here just because we know that this membrane is polarized, so it makes sense to include the thing that did the polarizing. And then I'm going to draw my little yellow hash marks to indicate that this center zone is no man's land. So if something is charged or polar, it cannot get through, and that includes ions, which is the primary thing that we are going to be discussing today. Ions moving across membranes. Okay, so let's label our major players here. So this is the sodium potassium ATPase. And this is going to move sodium out and let potassium in. And not only that, it's going to do it unevenly. So it's kicking 3 sodium out of the cell for every 2 it lets in. So every time this pump acts, we get a net loss of 1 positive charge. So because of that incongruity, the cell is constantly losing 1 more positive charge than it's gaining, which means we get a buildup of positive charge outside of the cell and less inside the cell. So now let's label our other players here. So this is going to be a sodium channel. So it's going to let sodium through. And this is going to be a potassium channel. And it's going to let potassium through. So now I'm going to draw a bunch of pluses on each side of the membrane. I'm not going to write out the whole ion name. I just want you to know that green pluses equal sodium and that blue pluses equal potassium. So that's the legend up here. So look at all those sodiums. And for good measure, let's put in a few in here, not very many. It's your membrane. You can do whatever you like with it. Just had a Bob Ross moment. Happy little pluses. So we have lots more sodium in here because via the sodium-potassium ATPase, the cell is sequestering potassium, and it's kicking out sodium. So right now, these channels are inactive. They can't do anything, which means that sodium wants to go out, but is denied because there aren't any channels open to carry it through. It's charged, and therefore it can't get through the yellow area. If it wants to go through, it needs some help. Same deal with sodium. It wants to go in, and nope, can't do. Also, this membrane is polarized because there is a lot more positive charge outside and a lot less positive charge inside, and there's different relative amounts of different ions on each side. So let's also make sure we put a couple potassium out here. So here we have a membrane that is at a resting potential, and that resting potential we're going to call -70 millivolts. But now we've introduced these players, the sodium and the potassium channels. So before I tell you what these do, I want to define some channel terms for you. So channels are transmembrane proteins that let things through. In this case, these channels are gated, which means they're not constantly open. They only open under certain circumstances. So channels can be gated in a variety of ways. So I'm going to switch colors here and give you some definitions for different kinds of gated channels. So the first kind I'm going to tell you about are ligand/chemically gated channels. And there are also voltage-gated channels. So ligand-gated channels are channels that open when the protein of the channel binds to a chemical. So open when bind to a ligand. And a ligand is just sort of a catch-all term for something that binds a large protein. So in ligand-gated channels, this could be a neurotransmitter. It could also be a hormone. Just some chemical binds to the protein, and that causes the protein to open and let ions through. The other is voltage-gated channels, and these only open within a particular voltage range. At all other voltages of the membrane, they're closed. I'm going to abbreviate particular voltage range. So these channels can open and let things through to depolarize or undo the difference between the two membrane sides, depending on what chemicals are present or what voltage the membrane is currently at. Now, typically ligand-gated and voltage-gated channels are not the same channel. So you can have a ligand-gated potassium channel that opens when a certain chemical hits it. You can also have a voltage-gated sodium channel that opens at a particular voltage, but those channels are not one and the same. They're different because their opening is mediated by a different thing happening. Okay, so let me draw you what happens when we undo the charge separation. So now it's about to get exciting. Okay, so let's say pink dots equal the neurotransmitter acetylcholine, which I'm going to abbreviate ACH. I'll write it down here. So you don't need to know any specifics about acetylcholine yet, just that it is a ligand. And I'm going to tell you that this is a ligand-gated sodium channel. So it's only going to open when acetylcholine is bound to it. So let's make that happen. So I have some little molecules of acetylcholine, and they are going to come bind to my ligand-gated ion channel, and then now my channel is open for business, and sodium ions are going to begin pouring through. So if I allow this process to proceed, what I'm going to end up with is a lot of sodium flowing in through this channel, but just at this particular spot of the membrane, because only this channel is open. Nothing over here is open. So that means that the other side of the membrane is going to lose a bunch of sodium. So this little chunk of membrane in this particular area has been depolarized locally. So over here, things are still in negative 70 millivolt land, but perhaps this area of membrane is now at negative 54 millivolts because I've undone some of the charge separation between the sides. I'm going to draw myself another voltage-gated sodium channel. So notice I said voltage-gated, meaning I'm changing the rules for it opening now. So this one is voltage-gated, meaning that it opens when membrane nearby it hits a certain voltage. Oops. So if this local depolarization proceeds to the extent that pluses from over here start flowing down their concentration gradients, voltage-gated sodium channels may open up. So let's say, for the sake of example, that instead of having this one be 54, now that we've moved some more pluses over, now this is negative 55 millivolts. Turns out voltage-gated sodium channels open at negative 55 millivolts. So we call this voltage the threshold voltage for opening, meaning it is not open unless negative 55 millivolts is reached by the membrane. So anything between negative 70 and negative 55, it's not open at, or anything below negative 70, it's not open at. It flips open only at the threshold voltage of negative 55. So now I've designated that, okay, the membrane has reached negative 55, so let's open our voltage-gated channel, meaning we're going to have an influx of sodium over here. So that's the difference in modality or working between ligand-gated and voltage-gated channels. Now notice so far we are only moving sodium, we are not moving potassium. So what if I want to redo the charge separation by letting some pluses back out? Well, I can open my voltage-gated potassium channel if I want, but my drawing's gotten pretty busy here, so I'm going to redraw this on the next page. Okay. So now I have my voltage-gated potassium channel sitting in the membrane here, and it is going to wait until the membrane has depolarized to its preferred threshold voltage, or its opening voltage, and that I will reveal to you shortly. In the meantime, let me draw my hydrophobic zone. And this is outside of the cell, and this is inside of the cell. So in the previous slide, what I did was made it so that there was lots and lots of sodium inside the cell. And there's probably still a little bit outside. And there's a couple potassiums out here. But there's a lot more potassium in here because I haven't opened any potassium channels yet. I've only opened sodium channels and let all those sodiums in. So in a cell with a resting membrane potential, which this is not currently, in a cell with a resting membrane potential, there is 70 millivolts less positive charge in here than there is out here. Now I've reversed that situation. So once a cell has depolarized, it actually goes past 0 and shoots up to positive 30 millivolts. So now there's 30 millivolts more positive charge inside of the cell than there is outside. So this membrane has been depolarized. So what if we want to repolarize it, get it back to negative 70, our resting potential? Well, we can do that by letting positive charge back out of the cell, but turns out this is a voltage-gated potassium channel, and it likes to be open at positive 30 millivolts. So once it detects that enough pluses have amassed in here, it will flip open. So let's open it up. And let all of our potassium out. And as the potassium leaves, the millivoltage is going to creep down. So as we get fewer pluses in here and more pluses outside, which I'm about to draw, the membrane will start dropping. So it'll drop to 10 millivolts and then maybe 10 millivolts and then maybe 5 millivolts and then 0 and then maybe negative 10. So we're going back towards the negative. So let's draw our potassiums out here. So the membrane I showed you just a minute ago, I said was depolarized because it was at plus 30 and there was a lot of pluses in here. Now that I've let some pluses out in the form of potassium, we call the membrane state something different. So now it is considered to be repolarized. And it actually repolarizes a little bit more than negative 70, so it goes down to closer to negative 90, and then it creeps back up. So what does it mean for it to creep back up? Well, turns out the voltage-gated potassium channel shuts once the membrane has repolarized and then hyperpolarized. Hyperpolarized means below the resting membrane potential. And then the sodium-potassium ATPase turns back on. So let's select our pink, which is our sodium-potassium ATPase color. So this turns back on and begins doing its job. So it will cleave ATP. So we have ATP being cleaved to a dp plus pi, and then we have the efflux of 3 sodium and the influx of 2 potassium. And once repolarization has occurred, this channel, because it's voltage gated and it only likes to be open at a certain time, will shut again. So all of the sodium-potassium ATPases in this membrane will get to work restoring that negative 70 millivolt resting membrane potential until the membrane returns to the state that it was in when I started this presentation. So what I'm going to do now is draw for you what that looks like on a millivolt over time chart. And this is a kind of chart that you'll see a lot because it's an easy way to summarize the voltage change curves for cells that are excitable and how they behave under certain conditions. So let me make myself some axes. Okay, and then I want a different color for this one. Let's do, actually, I changed my mind. We'll get to that in a minute. So let's label our axes. So on the y-axis is millivolts, and on the x-axis is time in milliseconds. So thousandth of a volt, thousandth of a second. So we have very small electrical changes over very tiny amounts of time. So this stuff happens fast. So let's label some relevant points on our graph. So this is negative 70. Here we have negative 55. Up here we have positive 30, and then maybe down here we have negative 90. So I'm going to make some keys over here in this legend. So legend, bam. So we're going to say that red equals resting membrane potential, and we know that the resting membrane potential is negative 70. So here we have a membrane chugging along at negative 70. And then when I start to have a local depolarization, probably mediated by some kind of ligand-gated ion channel, this is the local, also called the graded potential. This is the rapid depolarization that we call the action potential. But to stick with simplicity, let's just say that this is depolarization for now. So this is when the membrane is depolarized. Ran out of room. Ing. And I made it the same color as sodium. This line equals repolarizing. All right, so let's draw some stuff. So let's say my ligand-gated sodium channels open for some reason. Maybe the right chemical happens to be in the area and binding to them. And the membrane begins to depolarize, not particularly fast because it depends on the amount of hormone or neurotransmitter. But enough accumulates that it reaches negative 55. And negative 55 is our threshold voltage. So I'm going to denote with a dotted line here. So now that we've reached the threshold voltage, we get an actual depolarization. So this membrane is going to shoot up to positive 30. After depolarization, we get repolarization, so a rapid efflux of positive charge out of the cell in the form of potassium, and this is going to go all the way down below to about negative 90. So let's label which channels are open at which parts of this curve. So during resting membrane potential, the sodium-potassium pump, or ATPase, is active or open. So it's correct to say both. It's open and it's actively pumping. During the yellow part, that is ligand-gated sodium channels. That's supposed to be a U. I goofed. During depolarization, this is when voltage-gated sodium channels are open. But they shut at positive 30, making way for voltage-gated potassium channels. To open. Those close at about negative 90, and I'm going to add a line here. Pink represents the sodium-potassium pump, it's actively restoring the resting membrane potential. So this diagram is showing you changes in membrane potential over time. So remember, membrane potential means difference in charge and ions across a membrane. It's showing you which types of channels are open or closed at which times as well. So the closer we are to the plus side, if we go this way, so if the line is climbing, that means positive charge is entering the cell. If it's going in the opposite direction, that means that positive charge is leaving the cell. So I'll draw that too. I just need to erase real quick. So when the line is moving back down, that means positive charge is leaving the cell, either very quickly via via potassium channels, or more gradually. Okay, so that is action potentials and different kinds of ion channels, so depolarization and repolarization of the membrane front to back. And I want to remind you, for my classes, we are talking about this right now in the context of the neuromuscular junction and the sarcolemma. But I want you to know that this, of course, also occurs in neurons, which we haven't really gotten to yet. We've only talked about them in the context of the neuromuscular junction. But we will be looking at lots more of this kind of stuff as we move forward with our discussions of the body systems that include excitable cells. So don't think this is the last you'll see of this chart. You're going to see me draw it a lot more times in a lot of different ways. All right, and that sums up that. So thank you for your attention, and I will see you for the next video. Toodles!